Okay, hello, and thank you for being here today. We have our guest speaker, Dr. Zimmerman. He studies how the brain and body communicate with each other with a focus on understanding how feedback signals that influence learning and motivation systems in the brain. He received his PhD from UCSF in 2019, where his thesis research in Zachary Knight's lab focused on the body to brain systems that regulate thirst and drinking behaviors. He is currently a postdoc in Ilana Witten's lab at Princeton Neuroscience Institute. His postdoc work investigated how animals learn from ingestive feedback signals that arise from the gut after eating. For example, how we learn to dislike foods that make us sick. And in January of 2026, he will start his own lab in the Department of Neurobiology at the University of Utah. So congratulations to him for this new and exciting accomplishment. And without further ado, let's dive into learning more about his research into the brain and body interactions and learning.
All right. Thank you so much for that nice introduction, Ariana, and for the invitation to join you all today and tell you a little bit about my work. And from a high view, the goal of my research is to understand how sensory signals that arise from inside our bodies, from our internal organs, influence neural dynamics in our brains, and specifically how that contributes to cognitive processes like motivation and learning. Uh, but first, let's start with a cat video. Okay. So these cats are, of course, using sensory inputs to control their behavior here. And in blue, I'm just showing you all of the external senses that animals have. And I would say that most, if not all of these senses are engaged during this task. For example, the cats are using vision and audition to see and hear the bell, and they're using taste and smell to evaluate and enjoy these treats. It might be met less immediately obvious from watching this video, but I want to point out that the internal sensory environment of the cats also plays a really big role in regulating their behavior here. They're sensing that they're hungry, they're tracking how much they've eaten until they feel full or satiated, and they've learned through experience that these treats are safe and nutritious. And all of these components of their behavior are driven by interoceptive sensory systems. And interoception is really fascinating to me because while it's governed by a sensory system that's analogous to vision or hearing in many ways, it's also distinct in a lot of respects when compared to our external senses. Now, almost every organ in the body generates sensory signals, which encode everything from whole organism variables like energy and hydration levels, all the way down to the specific mechanical and chemical state of each organ. And these interoceptive signals are sent to the brain then through peripheral sensory fibers like the vagus nerve, as well as through the blood. And these signals are the nervous system's main tool for estimating the physiological state of the body. And this is really crucial information because, as we know, one of the ultimate objectives of animal behavior is to maintain a steady and healthy internal physiological environment that allows animals to survive. And so, a main goal in the interoception field today is to understand how exactly the brain connects this long and messy and honestly incomplete list of signals from the body to specific behavioral and cognitive processes in order to accomplish this goal. Now, for today, I just want to highlight that one key distinguishing feature between external and internal sensory signals is that they often operate on really different timescales. So while external stimuli offer the brain fast, real-time feedback on behavior, internal sensory feedback is often slow and delayed. And we can easily see this in the cats from that video I showed you. While the external stimuli in this task are fleeting, the internal consequences of eating will arise over slower timescales, from swallowing signals that are delayed by seconds to stomach stretch and intestinal nutrient signals that are delayed by several minutes to even slower signals from the bladder, colon, and fat reserves that can take hours, days, or even weeks to appear. And these slow timescales are a common feature of interoceptive signals, especially when we're talking about eating and drinking. But this long delay between action and feedback presents a big challenge for the brain. If the nervous system's main goal is to use these signals to control behavioral processes that are constantly updating in the moment. And so, a key goal of my work is to develop and apply new approaches that'll help us to understand how the brain solves these challenges. And so today I'm going to share with you two stories about the mechanisms that the brain has evolved in order to use these slow interoceptive signals to regulate fast behaviors like eating and drinking. First, I'll briefly talk about my PhD work where I studied how we're able to regulate thirst, so the motivation to find and drink water on a moment by moment basis despite slow feedback from our bloodstreams. And then I'll spend the bulk of my talk sharing some new work from my postdoc on a neural mechanism that allows us to learn from post-ingestive feedback from the gut that can arise after extremely long delays after a meal. All right, let's get started. So we experience thirst every day, but where does this sensation come from? Now, the classic answer to this question is that thirst is governed by the blood. And this belief was motivated by the concept of homeostasis, which was developed by the physiologist Walter Cannon in the 1920s and 30s. And as Cannon put it, homeostasis is the coordinated physiological processes which maintain most of the steady states of the body, involving the brain and the nerves and the internal organs all working cooperatively together. Now, the key here, the guiding idea is really steady states that thirst is a negative feedback process that maintains a constant hydration level in the body. And the components of the system then are really simple. We have an error signal that encodes dehydration in red. We have a detector in the brain that then receives this signal. And we have a behavioral output, so drinking water, that then restores this error signal to its proper set point. And this framework is so compelling that decades of research in the thirst field have really been focused on filling in the identities of these various components. So, for example, we know that dehydration signals are primarily embodied by an increase in the osmolarity or the saltiness of the blood. And similarly, pioneering work has shown that this dehydration signal is detected by neurons in a special brain region called the subfornical organ, or the SFO, that's conserved across all land vertebrates. And the SFO is special because it lies outside the blood-brain barrier, and it therefore has the unique ability to directly detect changes in blood osmolarity and other circulating signals that are important for regulating fluid balance. And electrical lesion studies and activation studies over the past several decades have shown that the SFO is necessary and sufficient to drive drinking in a whole range of species, including almost every mammal that's been tested. Now, the power of this negative feedback model is that it provides a simple mechanism to match our behavior on the right to our need levels on the left. But it also has a pretty critical limitation, which is that most of our normal drinking behavior is regulated on a fast, moment by moment basis that can't be explained by slow feedback from the blood. And this is actually true of almost all animals. And the classic example of this problem is the satiation or quenching of thirst. Now, when we're dehydrated, drinking water immediately quenches our feeling of thirst, even though that water won't begin to be absorbed into our bloodstream for tens of minutes. So this raises the question then of how the brain is able to force us to stop drinking on this faster time scale. As a grad student, I reasoned that, by recording the dynamics of thirst neurons in the SFO, which at that time hadn't been done before, might provide some insight into this question by allowing us to definitively determine what the inputs are that really regulate these cells. Now, to do this, we identify genetic markers like the gene NOS1 shown in red on the left that specifically label thirst-promoting SFO neurons, but not other intermingled cells in this tiny part of the hypothalamus. And to confirm then that this gene labels bona fide thirst neurons, we showed using optogenetics that this cell type is necessary and sufficient to promote drinking. So, for example, if we artificially activate these cells in mice that are fully hydrated, they aren't thirsty at all, it can cause them to go drink as much water as if they hadn't had access for days, as if they're completely thirsty. And on the other hand, if we inhibit these cells artificially in animals that really are dehydrated and should be feeling thirsty, they almost don't drink anything for a few minutes. And together, these results just made us confident that we're really targeting the correct set of cells. We're specifically labeling these thirst-promoting neurons. And so we next expressed a fluorescent reporter of neural activity, GCAMP, in these cells, and used fiber photometry to record their dynamics in freely behaving mice. And, as expected, dehydration robustly activated these cells. So the plot on the left shows that blood osmolarity was elevated by overnight water restriction and was then restored after mice were rehydrated with an hour of free access to water. And the plot on the right shows that the calcium dynamics of these SFO thirst neurons really perfectly follow these changes in blood osmolarity. They increase their activity when the mice are dehydrated, and this goes back to their sort of previous baseline level when the animals become rehydrated. And this is totally consistent with this classic negative feedback model for thirst that I showed you a few slides ago. So now I'd say we have the tools to start getting back to our original question of how the brain's able to rapidly regulate drinking, for example, by satiating our thirst, despite this slow and delayed feedback from the blood. Well, there's almost no change in blood osmolarity in the first 5 minutes after dehydrated mice start drinking. And I'm just showing you that in this sort of updated bar plot here. And so the negative feedback model clearly predicts then that thirst neuron activity, the spiking of these cells should be almost completely flat during the first 5 minutes of drinking. If there's no change in osmolarity, we shouldn't see any change in the activity of these neurons. And implicitly then, this model would tell us or suggest to us that this fast regulation of drinking must be happening somewhere else in the brain, somewhere downstream, like the cortex or the thalamus, etc. Now, on the other hand, it's possible that this model is wrong or incomplete, and that these cells also receive other inputs during behavior that help drive satiation. And to distinguish between these possibilities, on the right, I'm going to show you one of our very first recordings of thirst neuron dynamics. And in this recording, a dehydrated animal will get access to water, where the time zero, where the black line is. And every lick of water that that mouse drinks will then appear in blue. And I'll drink as well. No. Remarkably, the activity of these cells is not completely stable at all. And instead, these thirst neurons are rapidly inhibited each time the dehydrated mouse takes a drink of water. And this experiment really told us for the first time that these neurons must be regulated by some signals other than blood osmolarity, and that this simple negative feedback model really can't be the full story. Now, if we zoom out on the entire 60-minute experiment and all of our mice, we can see that this process has two clear phases. So first, these thirst neurons are rapidly inhibited during drinking, immediately after the mice get access to water. And then this inhibition is maintained. They stay inhibited after drinking ends, even though that water still hasn't been absorbed into the bloodstream. And the reason the finding that thirst neurons are inhibited by drinking was really exciting to us is because it might be able to explain how thirst is rapidly quenched before any feedback from the blood arrives. We just turn off our thirst neurons anticipatorily based on some set of signals in the world, and we then wanted to go in and understand what those signals are. Now, I could spend my whole talk on this question of what drives this inhibition, but to keep it brief, I'm only going to share two simple experiments that I think really helped us understand a lot about how this system is organized. Now, first, I want to show you what happens when, on different days, dehydrated mice drink pure water, which will restore their blood hydration and should quench their thirst, versus when they drink hypertonic salt water, which won't restore their blood hydration and, on the long term, shouldn't quench their thirst at all. Now, if we only look at the first minute of this experiment, we can see that during drinking, both of these liquids rapidly inhibit these thirst neurons to the same degree. And in this time, in this first minute, the volume consumed is almost exactly the same between these two groups. And follow-up work from our lab and from other labs showed that this inhibition during drinking is driven by a signal that originates from the mouth and throat and really precisely encodes the volume of fluid that the mouse consumes. Every lick produces an equal amount of inhibition, which is simply linearly integrated over time in the activity of these neurons. Now, but this mouth to brain signal, it's completely independent of fluid composition. It looks exactly the same for any liquid we could think of to test, from water to salt water to even oil. But we know from introspection and from a lot of decades of behavioral research that this volume signal alone can't fully explain how thirst is satiated because salt water won't rehydrate us. It won't quench our thirst. So how does the brain then know what we drank? Well, in the same experiment, we can see that over the next few minutes, thirst neuron activity rebounds to this previously elevated dehydrated level if the ingested fluid was hypertonic, whereas the inhibition is stabilized or even gets more profound if it's pure water. Now, based on the timing of this rebound effect, we thought that maybe this regulation is driven by a signal that arises from the GI tract. And so to test this idea directly, I teamed up with a postdoc in the night lab at the time, Lisa Beutler, to record thirst neuron activity while directly infusing liquids into the animal's stomach, now bypassing the mouth and throat altogether. And on the right, you can see the thirst neurons respond to a range of osmolarities of salt being infused into their stomachs, from pure water at the top to 500 millimolar sodium chloride at the bottom. And we can see that infusing water directly into the stomach rapidly inhibits these cells in blue, whereas infusing salt water, hypertonic salt water at least, rapidly activates them in red. And the reversal point of this effect is almost exactly at the osmolarity of properly hydrated blood, at the animal's set point. And this experiment showed us that there's also a gut-to-brain signal that precisely tracks not the volume, but the osmolarity of ingested fluids after drinking. And together, we thought that these new signals might provide a simple three-step mechanism for how drinking can immediately but persistently quench our thirst despite slow feedback from the blood. Now, first, blood signals initially tell the brain how dehydrated we are and activate our thirst neurons proportionally. Second, mouth to brain inhibit these thirst neurons during drinking, and they basically tell us when we've drunk enough to restore our deficit. And then later, these gut to brain signals tell us whether what we drank was actually, in fact, water and stabilize this inhibition of thirst neurons if it was. This was super cool for us. And finally, just one more thing in this vein, we wondered if this mechanism that I just described to you might be carried out within individual neurons in this circuit. So previously I was using fiber photometry for all of these experiments I showed you, so we couldn't really disentangle individual cells. Or alternatively, if it's somehow distributed across this population of neurons in some way. And to test this, we used miniature microscopes to record the activity of individual neurons in the brain's thirst circuit, and to ask whether the same cells or different cells receive each of these fast and slow sensory signals. And in these heat maps, neurons are registered, they're aligned horizontally across experiments. And you can see that the answer to this question is really a simple yes. Each of these cells is activated by dehydration, is rapidly inhibited by drinking water, and is also inhibited by direct infusion of water into the stomach. And so this further tells us then that individual thirst neurons are able to integrate fast predictive signals from the mouth and gut with slow hydration signals from the blood, which should allow them then to calculate the body's need for water in real time and to start and stop drinking accordingly. Now, to summarize this first part of my talk, a major challenge in this field was that the brain's thirst system for decades was thought to only be regulated by negative feedback signals from the blood. But these signals are too slow to explain most normal drinking behavior, which evolves on a rapid moment-by-moment basis. And we discovered that the brain solves this timescales problem by using faster sensory signals that arise first from the mouth and then from the gut during drinking. And that together, these fast interoceptive signals give the thirst system a shortcut to anticipate delayed feedback from the blood and adjust drinking behavior preemptively. And as a grad student, this was just an incredibly fun journey that really opened my eyes to how much we still have left to learn about the sensory systems within our bodies, and especially their influence on the brain and on our behavior. And this really convinced me to continue exploring this system during my postdoc, and in my own lab as well. And so next, I'd like to tell you about a different challenge for the brain that results from these slow timescales of interoceptive signals. This time focusing on the problem of learning across long delays after a meal ends. Now, as we know, animals learn from experience, and neuroscientists often study how animals learn the value of cues like sights or sounds or of actions based on rewarding or aversive feedback. Now, for example, over time, this mouse will learn that the color and shape of blueberries and the sight of the blueberry bush predict their delicious taste. And this trial and error learning process unfolds over, seconds or even milliseconds. And I'd say that we have a pretty good understanding of the mechanisms for credit assignment. That is, associating the feedback signal to the correct stimulus out of all the possibilities in the world on this short time scale. However, there's another time scale of learning that could be at play in this photo. And that's because after eating, signals from the gut tell the brain whether foods were toxic or safe. And these signals can change our preferences for those foods through one-shot learning, so after just a single experience, even though the feedback arrives minutes or even hours after a meal has ended. And in the real world, this is what happens when we get food poisoning. However, as a field, we know very little about how the brain is able to bridge this long delay to support learning. And at least for me, this is a really interesting puzzle because the mechanisms for credit assignment on short time scales probably aren't relevant here. Because in general, they rely on a neural representation of the stimulus that overlaps in time in the brain with the feedback signal. So, for example, just to simplify this, like the mouse in the previous picture could see and smell the blueberry bush while, or at least immediately before it was eating those berries. But a mechanism like that would be pretty surprising with delays of minutes or even hours. And so during my postdoc, we used a classic conditioned taste aversion or a CTA paradigm to study this problem. And in CTA, mice are allowed to consume a tasty drink, sweetened Kool-Aid, and then 30 minutes later, they're given an injection of the drug lithium chloride to produce a visceral malaise that's pretty similar to food poisoning. And 2 days later, we can then use a two-bottle preference test between the paired flavor and water to measure learning. Now, our entry point in studying this problem is the fact that mice only learn in this paradigm if the paired flavor is novel to them, if they've never experienced it before, which is what I'm showing in the middle here. So the lithium chloride group has developed an aversion to this flavor after only a single pairing with delayed food poisoning symptoms, whereas the control group in black that didn't experience any illness and just got a saline injection still loves this sweet flavor. However, if the exact same flavor is familiar and it's known to be safe, then there's absolutely no learning in this paradigm, which I'm showing on the right. And here, the familiar group just had several days of Kool-Aid access without any negative consequences before the lithium chloride conditioning day. This is a pretty crazy effect. To be clear, this is only true in the case of one-shot learning. If you repeatedly make these mice for which the flavor is familiar sick, and you do that over many days, they will eventually learn, but they can't do it after a single experience. And so our goal was to use this behavioral difference between novel and familiar flavors as a window into understanding how this extreme learning process plays out in the brain. No. First, we needed to know where in the brain the signals for flavor novelty and for visceral malaise converge, because this could be an important site for the plasticity that underlies learning. And to accomplish this, we mapped neural activation across the entire brain at all three stages of our CTA paradigm. So immediately after flavor consumption, later during delayed malaise as the animals are feeling sick, and then days later during memory retrieval when we force them to recall this memory. And we did this for mice for which the flavor was novel, so they should learn, as well as for control groups for which the same flavor was familiar, and so they shouldn't learn. We used light sheet imaging of intact cleared brains to generate high resolution 3D reconstructions of endogenous FOS expression as a proxy for neural activation for each of these animals. We developed a new deep learning assisted cell detection pipeline to identify all of the activated neurons in this huge data set and then mapped each of these cells to the Allen Institute's Mouse Brain Atlas. And finally, we created some new statistical tools to specifically estimate the effect of flavor novelty on activation in each brain region at each stage of this paradigm across many cohorts of mice. And this produced a really dense and, I would say very interesting data set. But I'm only going to talk about one finding from it today. But I do want to point out that, in parallel, we developed in collaboration with Sam Wong's lab at Princeton and David Kleinfeld's lab at UCSD and a few others, an online interactive visualization for browsing all of the 25 million activated neurons in this data set. And I just point this out, even though you may not be interested in this specific experiment, because I think it gives us a nice roadmap for how we can make these large brain-wide imaging data sets more useful to the community and more shareable, because this is really becoming a basic part of how we do neuroscience. And I think it's important that we get as much as we can out of these rich data sets. But for today, the plot on the left really gets at the story I want to tell you about. So in this heat map, each column just represents one time point of our paradigm. So when the mice are consuming the flavor first, later when they're feeling ill during malaise second, and finally when they retrieve that memory all the way on the right. And each row corresponds to one region of the mouse brain. And the color of each cell indicates how strongly that neuron or how preferentially that neuron was activated if the mouse ate something familiar in blue before they felt sick versus when they ate something the same flavor when it was novel in red before it was sick. So a red cell indicates that that brain region was much more activated in the mice that ate something novel when compared to the mice that ate something familiar. And we're then just sorting all of these brain regions based on similar activity patterns with a simple clustering algorithm. Now, there's a lot here, but what really jumped out at us was the cluster of regions shown at the top that was preferentially activated by novel flavors that promote learning at every stage of this CTA paradigm. And this cluster almost exclusively consisted of subregions of the amygdala, especially the central amygdala or the CEA. And this was really exciting to us for two reasons. So first, the fact that a novel flavor representation that was initially formed during consumption, during a meal, might still be present in the brain 30 minutes later suggests that this network might be the site for the convergence of flavor and malaise signals. And this is exactly what we had been looking for at the outset of this project. And second, the fact that this representation persists when the memory is retrieved days later in the same set of brain regions suggests that the amygdala might also be a site of storage and recall. Now, the amygdala is also a strong candidate for bridging this long delay, not only because half a century of work has consistently implicated it in many forms of associative learning, but also because we know that the amygdala directly receives visceral malaise signals from the gut via projections of a population of cells in the hindbrain called CGRP neurons. Now, CGRP is a neuropeptide that these cells co-release, but they're glutamatergic neurons, and for today, we'll just think of them as excitatory cells. Now, these neurons were initially discovered and characterized by Richard Palmiter's group, who had previously showed that these CGRP neurons are necessary and sufficient for relaying malaise signals from the gut to the forebrain for CTA learning. So just to make that more clear or more concrete, we found, for example, that CGRP neurons are activated by malaise induced by a lithium chloride injection in vivo, which I'm just showing here with a fiber photometry recording. We also found that, like lithium chloride, delayed optogenetic activation of these neurons, these CGRP neurons, after a meal can condition a taste aversion to novel flavors, but not to familiar flavors. And we also saw that stimulation of these cells more strongly activates the amygdala when that stimulation was preceded by consumption of a novel flavor when compared to a familiar flavor. And this is again just like lithium chloride. And critically, this CGRP neuron pathway could also give us, as experimenters, a tool for testing with high temporal precision how malaise signals impact neural dynamics in the amygdala during CTA. So these brain-wide FOS measurements suggested to us then that the amygdala is where novel flavor and malaise signals converge to support CTA learning. But this still leaves open the really interesting and informative question of how these temporally separated signals are integrated at the level of the individual neurons in this brain region. No. One hypothesis is that novel flavor coding neurons, but not other neurons in this part of the brain, are persistently activated for long periods after a meal end. And that this then provides passive overlap with CGRP neuron malaise signals and allows plasticity to occur. Now, this would be analogous to taking typical mechanisms for credit assignment on shorter timescales in trial and error reinforcement learning tasks and just kind of stretching them out to accommodate the crazy delay that we're dealing with here. Now, alternatively, CGRP signals might specifically reactivate novel flavor coding neurons, but not other cells. And finally, CGRP signals could also activate a completely separate or only partially overlapping population of amygdala neurons that then subsequently become incorporated into that novel flavors representation during a memory consolidation process. Now, each of these mechanisms could explain our FOSS data, and they could support learning. But to distinguish among them, we needed to track the activity of individual neurons across the stages of this CTA paradigm. But we obviously couldn't do this with FOSS because it's limited to only one or maybe two measurements per animal. And so we developed a strategy for chronic high-density electrical recordings in the amygdala, combined with optogenetic stimulation of CGRP neurons in the hindbrain in freely behaving mice. And here you can just see a reconstruction from one mouse of a four-shank neuropixels probe trajectory overlaid on the Allen atlas, with the full shanks in the brain shown on the left, and only our active recording sites in the central amygdala shown on the right. And when I overlay all of the animals in the main experiment, you can see that we can really precisely target the CEA using this strategy. We then trained these mice through a variant of our CTA paradigm that had just two key changes. So first, during drinking, there are now two reward ports, one containing the novel flavor, just like before, and then another containing water as an internal control for satiety, movement, the act of drinking, and reward. And second, the mice were moved to a distinct second context immediately after consumption ended, which just ensured that any neural correlates of flavor or drinking that we might subsequently observe in our recordings weren't due to features of that original context where consumption occurred. And after a 30-minute delay period in this second context, these mice then received 45 minutes of intermittent CGRP neuron stimulation to create basically a virtual food poisoning state in their brains. We recorded something like 1,000 central amygdala neurons in this paradigm. And the first thing we did was simply classify each cell as novel flavor or water preferring, or neither, during consumption. And here, every neuron's drinking response is shown to the novel flavor on the left and to the water on the right. And we then asked how these three groups of neurons, or these three subpopulations of neurons in this brain region, react over the remainder of this experiment. And you can see, for example, that each of these groups had similar activity levels overall during the initial consumption period. Now, the first thing we noticed is that after consumption ended, each group of neurons began firing much less. And this is obviously inconsistent with the credit assignment mechanism based on some special persistent activity of novel flavor neurons, as in the first hypothesis I laid out. And so we can cross that one out for now. Okay, so the real key then to distinguishing other mechanisms we've been talking about is what happens when we begin to activate these malaise promoting CGRP neurons. Now, remarkably, stimulating these CGRP neurons in the hindbrain during this green period in this plot strongly and selectively reactivated the same neurons that encoded the recently consumed novel flavor, but didn't really have as much of an impact on the other groups of cells. And this is only consistent with the second hypothesis. And zooming in then on individual 3-second bouts of CGRP stimulation, we can confirm that this reactivation appears to be driven by fast glutamatergic input from CGRP neurons. You can see these rapid time-locked responses to the laser. That's stronger for novel flavor encoding neurons than for other cells. And I actually think this is a really important control because it tells us that this isn't, alternatively driven by some slower change in physiological or emotional or endocrine state that builds up during this long period of virtual illness. No. The population dynamics from these recordings tell a really similar story. At the population level, activity during consumption of the novel flavor and water were highly distinct. And here I'm just showing you the neural trajectories for each of these experiences in a two-dimensional subspace from performing PCA across all of our recorded neurons from all of our experiments. And we can see that the second principal component, PC2, so the vertical axis here, perfectly discriminates these two experiences. Now, projecting the population dynamics during CGRP stimulation into the same subspace revealed a remarkably similar trajectory to the experience of consuming the novel flavor, which again is consistent with the single neuron results I showed you a moment ago, suggesting that CGRP neuron stimulation in illness can somehow cause the brain to revisit the experience or the representation of a novel flavor from a recent meal. Okay. So we wondered then, do these flavor reactivations also occur during normal physiological malaise where we're not using optogenetics to do this sort of fancier experiment? And to test this, we repeated our recordings using a lithium chloride injection instead of CGRP neuron stimulation. And once again, malaise strongly and selectively reactivated novel flavor coding neurons, but not other amygdala neurons. Then in a separate cohort of mice, we ablated CGRP neurons using a genetically encoded caspase to test whether these cells are really absolutely required for this effect. And in these animals, lithium chloride injection didn't reactivate novel flavor neurons at all. I mean, it didn't really have much of an effect on the central amygdala, which makes sense given that this is the main set of neurons that are conveying illness signals to the forebrain, and that their main projection is to the CEA, and we've completely obliterated them. But it's still really reassuring to see. And so together, these recordings showed that delayed illness signals selectively reactivate the amygdala's representation of recent novel flavors, and that CGRP neurons in the hindbrain are necessary and sufficient for this effect. Now, this was super intriguing to us, but what we really wanted to know then is if these malaise-driven flavor reactivations, causing the animal to revisit what it ate during a recent meal, if this might serve to stabilize or to strengthen that flavor's representation when that memory is later recalled, like our simple hypothesis here suggests, but also which would be important for this playing a meaningful role in learning. And so to test this, we took advantage of the stability of our chronic neuropixels recordings to track individual neurons across conditioning and retrieval days using their electrophysiological signatures. And here I'm just showing you these signatures for one particularly beautiful example neuron on 2 consecutive recording days. Now, dividing the novel flavor neurons based on how strongly they were activated by CGRP neuron malaise signals on conditioning day. So dividing cells based on how strongly they felt the effects of food poisoning, you might say, revealed a pretty striking difference. The amygdala neurons with the strongest CGRP input in the middle here responded much more intensely to the paired flavor when the memory was recalled. Whereas the remaining neurons had a slight decrease, if anything, in the response. They didn't really change very much. And we can also see this effect by simply plotting the change across days in each neuron's response to the paired flavor on the y-axis in this plot. So this is just showing what the response was on the retrieval day or the recall day, minus what the response was on the conditioning or the original day. And looking at this as a function of that neuron's response to CGRP neuron stimulation on the conditioning day, which is on the x-axis. And we can just see a nice linear trend here. And this suggests then that CGRP activation during illness triggers plasticity in novel flavor coding neurons that then strengthens the amygdala's representation of that potentially poisonous flavor after learning. Now, we next tracked individual cells as mice consumed a novel flavor, but this time without any aversive consequences. And then a few days later, consumed the same flavor again when it was familiar. And here we saw really different results. So the flavor's representation was significantly degraded and became more similar to that of pure water after it became familiar. We can see this basically any way we look at the data. And what I'm showing you here is just that the neurons that responded intensely to that flavor when it was novel respond much less intensely when it's familiar a few days later in blue. And together, these multi-day experiments suggested that illness signals induce plasticity in the amygdala to strengthen the representation of recent flavors that might be poisonous or might have been poisonous. Whereas flavor representations rapidly degrade in the absence of that feedback as flavors become familiar and safe. And altogether, we think that these findings provide a nice starting point for a mechanism that might allow us to learn from post-ingestive feedback, even after really extreme delays. First, our brain-wide FOSS screen showed that a network of amygdala regions is unique in being preferentially activated by novel flavors that support learning across every stage of conditioned taste aversion. Next, our chronic neuropixels recordings showed that post-ingestive illness signals selectively reactivate the amygdala's representation of recent novel flavors, and that this is mediated by hindbrain CGRP neurons. And finally, we showed that these post-ingestive reactivations induce plasticity or change the response properties of these cells to stabilize or strengthen the overall representation of that flavor in the amygdala after learning. And at a broader level, one of the reasons that this is really exciting to me is that it could provide a general framework for one-shot learning across long delays. And this model really just has two simple parts. First, some dedicated circuitry for detecting and tagging novel or salient experiences, here played by sensory or limbic regions. And second, built-in priors about possible stimulus outcome associations that exist in the world. For example, like selective reactivation of flavor representations by illness signals, but not by other kinds of feedback signals. And I really like this framework for a few reasons. First, it suggests that the common phrase that we associate with unexpected nausea, that it must be something I ate, is literally wired into the brain in the form of this evolutionarily built-in prior. And second, because looking beyond conditioned taste aversion, one-shot learning across long delays is a common challenge in the real world that this model could help to simplify by dramatically reducing the dimensionality of stimuli that are eligible for association in any given situation, simply by taking advantage of knowledge about which stimulus outcome associations are more likely based on evolutionary experiences of the entire animal kingdom. And personally, this framework also lays out some really interesting questions about how this algorithmic or systems level mechanism is implemented at other scales. And so for the last few minutes of my talk, I'd like to share some ongoing work and specific projects that really get at this idea. One key question, for example, from the model I just shared with you is how malaise signals or illness signals are able to correctly reactivate the representation of a recent novel flavor, but not all of the other stimuli of various modalities that are encoded in the amygdala. Now, our thinking was that there might be a biochemical tag that persists in novel flavor neurons after a meal ends, and that this is then important for deciding which cells are reactivated by malaise signals and which are not. And this idea builds on work from Alcino Silva, Sheena Jocelyn, Denise Kai, and many others, showing that increased excitability helps to recruit neurons into a memory engram in a lot of learning paradigms, including in CTA. And that protein kinase A and its substrate CREB are important for this process. You know, our idea was that maybe novel flavor consumption turns on this signaling pathway in the amygdala, and that this then leads to novel flavor neurons being more excitable for long periods after a meal ends. Now, to test the first part of this idea, that novel flavors might preferentially trigger PKA activity in the amygdala, we monitored PKA dynamics in freely moving mice with fiber photometry recordings of the new sensor ACAR2. And we did this as mice either consumed a novel flavor or a familiar flavor, or for comparison, water. And the short answer to this question, again, is yes. Novel flavors strongly trigger PKA activity in the amygdala in a manner that outlasts spiking by tens of seconds after each feeding bout, whereas familiar flavors or water have small or no effects. And this is consistent then with the idea that novel flavors may create a biochemical trace in the form of PKA and CREB activity and their downstream effects on excitability that leads to selective reactivation and plasticity of novel flavor neurons when compared to neurons or synapses encoding other flavors by these delayed illness signals. And so, to be very concrete, the idea here is that you have some wiring specificity of illness inputs from CGRP neurons onto all of the neurons in the amygdala that encode flavors, but not other stimuli. And that gives us stimulus outcome specificity. And that this model then helps us to arbitrate between different possible flavors that you might learn about. That the synapses you choose to potentiate are selected by this mechanism, which gives you only the novel flavor representation being boosted, but other flavor representations being left relatively intact or even degrading. And I'll just point out that I think this model is exciting in part because it really expands previous ideas for how PKA and CREB contribute to learning. First, it suggests that these increases in excitability that bias neurons into a memory can be actively triggered. They're not just some passive and stochastic property of the neural population in every system. And second, it suggests that the eligibility time window for learning after a meal, which is like the thing that actually makes this behavior and this learning paradigm unique when compared to many forms of learning we study in the lab, that this time window might be directly encoded in the slow decay to baseline of this increased excitability. And this might then give us in a window into picking apart what actually determines that eligibility window. Now, the data I showed you here is really obviously just a first step, and moving forward, some of the first projects in my own lab will be to directly test this model. With that, I'd like to thank everyone who contributed to these projects. My earlier work on thirst was done in Zach Knight's lab at UCSF. And my recent work on conditioned taste aversion was done during my postdoc in Ilana Witten's lab at Princeton. Both have been just fantastic environments to train and to do research. And of course, I'd like to thank you for your time and for the invitation to share this with you. And as one last thing, I'll just do the plug that I'm starting my lab in January at the University of Utah. And if this work sounds interesting to you or to someone you know, I'll be recruiting students and postdocs, so please reach out. And with that, I'm happy to take any questions that you have.
All right, thank you. That was so interesting. I'm going to start this off with the first question, and then anyone can ask their questions after. Mine is a little lengthy. So most research on credit assignment focuses on short delay learning, yet this study examines how flavor associations form across much longer temporal gaps in post-ingestive paradigms. So how might investigating learning over extended delays provide a deeper insight into the mechanisms of credit assignment overall? And then this question might be for anyone, but I wonder what kind of behavioral or experimental paradigms could be used to study long delay learning beyond post-ingestive feedback. Yeah.
Yep. So to answer your first question about what we might learn more deeply about learning or about credit assignment from studying this problem, I think, there are two answers to this. One I kind of laid out at the end of my talk, or near the end, which is that learning across long delays is a problem that we don't really study in the lab. And we do this, you can think of examples in your life where you have, back pain and you may think about all of the physical activities you did that day. Or you may arrive at the wrong destination when you're trying to navigate there and think back about all the turns you took along the route. And I think there's a really strong analogy to what we're finding with conditioned taste aversion, which is that there's some prior you have based on the feedback signal, whether that's getting to the wrong location and thinking about spatial decisions, or feeling pain and thinking about physical exertions, that at least in the human world, just going by introspection and talking with people, that involve or may involve something really similar in terms of some hardwired prior, like an idea that this kind of outcome is probably a result of this class of decisions I made or stimuli I encountered, and that the brain might take advantage of that. It'd be really smart to take advantage of that in a way that lets us learn. Because if you try to learn across a long delay, the key challenge is that we face like an ever increasing bombardment of stimuli and make an increasing number of actions every second that goes on. And if you think any stimulus or action is eligible for learning, it's going to make your life really challenging. It's going to make this learning problem really challenging for the brain to compute properly. And so this just simplifies that idea. And so the hope, is that we can take these ideas and apply them to other forms of learning in the laboratory, or take them to, studies of humans doing various tasks to ask if this might be true. But that's the analogy that I draw in terms of what we might learn about this more broadly. Um, and I think, just going a step further, this idea of hardwiring stimulus outcome associations, it doesn't need to be limited to long delays. It could be something that happens on shorter timescales as well. And I think there's some evidence for this even in RL tasks, right? Like it seems like representations or neural correlates of learning emerge more quickly, for example, in a visual learning task in parts of the striatum that receive a lot of visual input from visual cortices. And you might imagine that this is a more general property of the striatum, or some parts of the brain that allow stimulus outcome associations in a preferential way, that we're not just some flexible clean slate.
Thank you. And then I think Emma has a question over Zoom.
Hi, yeah, thank you for your talk. That was super interesting. I just have a question with regards to what you think the circuitry or the pathways would look like if the feedback is responding to something that is, rather than inducing malaise, something that is highly nutritious, something that is the interoceptive response is one that is positive, because obviously in the amygdala responds to novelty, rather, like whether it's positive or negative. Um, do you think that there is perhaps like a different pathway also through the amygdala that is encoding a positive response, like a highly nutritious food?
Yeah, this is a fantastic question. So we know from a lot of behavioral work from the 80s and 90s, that started in Anthony Sclafani's lab, that this kind of learning does occur. So if you pair a flavor with infusion of different macronutrients into the stomach, rats will learn to prefer those. And there's evidence that a similar process is at play in humans. And over the last few years, we've learned a pretty reasonable amount actually about the pathways that bring that information into the brain. The common theme of this is, it's always a little bit messy, but the common theme is that the vagus nerve detects these signals, and that the main consequence is the release of dopamine in the forebrain. Different groups have done different experiments and had different findings, with some focusing on dopamine release in the striatum, and others focusing on dopamine release in the basolateral amygdala. And I think, this suggests to your point then that at least some people are seeing a huge response in the BLA, like an enormous response in dopamine to nutrients in the gut, that there could be a similar process at play for learning associations with nutrients, just in a different compartment of the amygdala. Um, but I think, it's a really interesting problem to me, at least, because I think the way credit assignment is implemented, the way this is influencing the downstream neurons in the amygdala or in the striatum is probably different than conditioned taste aversion. And the reason I say that is that, for one, we're already invoking dopamine as a neuromodulator, which we think of as driving plasticity, as opposed to this like glutamatergic signal coming in when you're feeling sick. But also, as far as we can tell, this operates on a much shorter time scale. Like, there's no crazy careful study of what delays can be supported by nutrient detection. But with, pure sugar diets, these feedback signals can arise as quickly as like a minute later, or even faster. And so I think that combination of a shorter time scale and dopamine makes me think that we might be playing with a credit assignment mechanism that's more similar to what we think about in like RL tasks, but just, stretched out a little bit to accommodate this, as opposed to this like totally bespoke solution to account the fact that food poisoning can arise like a day later, and still allow animals to learn. But a lot of that's still speculation.
Thank you.
Okay, we have some other questions here. Who wants to go first?
Yeah, my first one was applying some of the ideas from the second portion of the first portion of the talk. Fantastic talk, by the way, Chris. If you want to build an expectation of the hydration of a given liquid, I assume you'd want access to the rate of ingestion and the osmolarity in the gut after. So do you know if the neurons in the SFO, has anyone followed up to see if those signals within the SFO neurons are used to build a representation or update expectations of hydration for familiar or novel liquids? Or is it
Yeah, so I think there's been some work showing that manipulating SFO neurons. So I guess as a first point, I'll say like SFO neurons, as far as we can tell, encode to some extent this prediction based on sensory feedback during ingestion. They're not really good as, at least we have no evidence that they can predict. This seems to be hardwired, right? That you count licks and then you count molecules of salt in your gut and that you put that information together. But if they've learned over time that something is salty, that doesn't impact, for example, this like early inhibition of these cells. So it really truly, as far as we can tell, is feedback and isn't predictive in the sense of involving learning in the adult in the SFO. And so I think that makes it might provide a teaching signal for a downstream region to construct the kind of representation you're talking about, where we like, for example, if I'm thirsty and I encounter something that I've learned won't rehydrate me, that I don't want to like release much dopamine in response to that, to like oversimplify matters. But I don't think that sort of careful test has been done. But a prediction would be that that's a good way to use this signal.
Thanks. The second quick one was that you showed those beautiful maps of activity. And you were focusing on the central amygdala, but I couldn't help but notice that the lateral and basal nuclei seem to have opposite responses to familiar and novel. And part of the reason that stuck out to me is because one-shot olfactory aversive conditioning is mediated by the basal nucleus, but most of the Krebs-mediated engram construction work has been done in the lateral nucleus. So I'm wondering if you think like are CGRP receptors somehow driving the activity differentially across these nuclei? And if you repeatedly present the familiar flavor and drive an association, are you changing activity in the lateral, or are you somehow engaging the basal nucleus newly?
Yeah, there are a few parts to this. So, So we see that the BLA, parts of the lateral amygdala, parts of, the intercalated amygdala, which is this like little rando chunk right below the CEA, all have like relatively similar response properties and that you can like decode with something was novel or familiar. Um, how those are all interrelated and what the source of them is a great question that is something I'm excited to work on moving forward. But right now we don't have great information. One thing I'll say is that, one possibility is that it's inherited from cortical regions because at least during consumption, we see this same effect in piriform, like of preferential activation by novel flavors in piriform and in the insula. So one possibility is that it's inherited. And another possibility, obviously, is that this novelty signal or familiarity signal, depending on the direction we're talking, is implemented with some neuromodulator that's broadly accessible to these circuits. But I think there's a lot of important questions about what different parts of the amygdala are actually doing here, because it's presumably not a wholly redundant system the way I've kind of been treating it today.
Then Kathy has the question.
It seems like another layer where that might allow you to get plasticity and familiar is odor, right? Because if a food's gone bad or something like that, it seems like a really obvious factor, sensory stimulus to add in, and you might get a change in the familiar odor.
Yeah, so I think the contribution to odor to this is like much more surprising than I anticipated it would be when I like came into this field. So I'll say that the experiments I've done we're using sweetened Kool-Aid, so it has an odor component and it has a flavor and a taste component. The classic behavioral work I do think it suggests exactly what you're saying, that odor is a key component, but with a few caveats layered onto that. So animals really struggle to learn in the absence of a taste, like in the absence of a basic tastant being present. Which seems to be like a gate for learning, for example. Like you need a taste to be present, otherwise we can't learn in this long delay situation. But then all kinds of useful second order conditioning can happen if that taste is present, and the most strong seems to be onto odors. So, it seems to be a process where the animals are learning an odor taste association and then a taste outcome association. There's a lot of like devaluation work that makes us think that's what's happening. But it suggests that, you're right, odor is like arguably a better indicator. It's this, and ethologically, it's the signal you would want to use because you don't have to actually consume anything to take advantage of it. You just sniff something. And it also suggests that this is probably like a second component of this learning process that might be even happening somewhere else in the brain. You can imagine conditioning odor taste outcomes or odor taste associations in cortical regions, and then the basic taste to illness association subcortically. But like how that works in the brain is all speculation right now, but it's a really interesting area.
I was especially thinking about odor where food has gone bad, right?
Yeah, yeah. You mean like being predisposed by some particular odor?
Yeah.
Yeah. I mean, it's a great idea, but to my knowledge, there hasn't been a ton of work on it, but I agree. It seems like it would be an obvious indicator to use, at least in species that have food sources that smell off in a predictable way, like we do, but I don't know like how far back that goes evolutionarily.
Brad had a question. Maybe
Do you have a question?
I did, but we're after two.
it's
Quick, naive question. When I learned about this stuff back in the day, this was called the Garcia effect. Is that no longer a thing?
It is, still commonly called the Garcia effect. The seminal work was done by, John Garcia when he was a researcher at a naval research laboratory at UCLA and several other places. But there have been great contributions from other people since then. So, I, for nomenclature purposes, just went with conditioned taste aversion, which is more common now, but it's effectively the same behavioral paradigm as the Garcia effect. There are some differences, obviously, like Garcia's initial experiments involved irradiating rats, which is not how we make them feel sick anymore. So there are some differences, but this like long delay phenomenon, totally the right name.
All right, thank you.
All right, thank you so much.
Thank you.
Very elegant talk.
Yeah, that
Thank
was
you. That was
amazing.
really nice.
Very interesting. Yeah.
Thank you.
Thank you. Thank you.
Ollie, are you meeting with?
Yes, yes, we're meeting later, right?
Yep.
See you.
All right. See you later. Goodbye,
Yep.
everybody.
Bye.